division or what kind of division? Reductional or equation?
Equational, right?
Okay. Why? You are right. Why?
Because the number of uh, chromosomes in the parent and the daughter cells remain same.
Very good. Meaning if the parent, initially, if the parent was diploid, then the offspring would also be diploid, isn't it?
Yes, ma'am.
Okay. Now, another thing, yes, listen, I have gotten to know about the tissue chapter and that structural organization in animals. And in that particular chapter, many people are saying that the different types of tissues are going to come. Have you confirmed it from anywhere? Because I have taught you the whole entire frog, but the cockroach, along with the 4 different types of tissues, they will also come. Have you confirmed this?
Ma'am, I have a PDF of
Hmm.
the final portion for NEET 2024, and that it was not mentioned. So that's what I told you also in that class.
Like, okay, it's fine. But there is another NEET student who I teach, and she told that it is coming. I don't understand what is the case
Oh,
now. If you are saying that, what I was thinking, like once we are done with the whole entire syllabus, the parts that are 100% coming, I think I'm going to teach you that. We can never be like too prepared for NEET, isn't it?
okay,
So
ma'am.
when like all the chapters are done and we are completed with the syllabus, I will teach you, like give you a brief so that even if the question comes, you will be able to answer it. Okay.
Okay, ma'am.
All right. Give me 1 second. All right. Okay. So to begin with, now we're going to discuss about cytokinesis. Now, always remember that the karyokinesis, whenever it is done, okay, in normal situations, the karyokinesis will be followed by the cytokinesis always. Now, karyokinesis was the division of the nucleus. Now, cytokinesis is finally when the nucleus has divided, now the cell will divide. So talking about cytokinesis, so mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei. Two nuclei are present in one cell, but now the cell itself is divided into two daughter cells by the separation of cytoplasm. And this separation or division of cytoplasm is known as cytokinesis. And it happens at the end of every karyokinesis. Now, in an animal cell, this is achieved by the appearance of a furrow. Now, see, the cytokinesis differs in animals and plants both. Talking about the animal cell cytokinesis, so it happens when a furrow is formed, sort of deepening of the cell starts take place and then it moves towards the outside, dividing the cell into two. So the deepening or the furrowing in the plasma membrane takes place. Now the furrow gradually deepens and ultimately joins in the center, dividing the cytoplasm into two parts. Plant cell there is no furrow formation, rather in the plant cell it takes place by the formation of a cell plate. Plant cells, however, are enclosed by a relatively inextensible cell wall because animals do not have a cell wall and plants do. So their way of cytokinesis will be much more different as compared to the animals one. So therefore, they undergo cytokinesis by a different mechanism. Now, in plant cells, wall formation starts in the center of the cell and gradually and grows outwards to meet the existing lateral walls. Now, one thing to remember, do you people have you studied in physics when the force is applied from the outside to the inside, what is that called? And when it is applied from the inside to the outside, what is that called? Have you people learned this? Centrifugal and centripetal? Hmm? Say yes or no.
Yes, ma'am.
Have you learned this? Tell me which one. Centripetal is from outside to inside or centrifugal is from outside to inside? See, just to see the difference between the cytokinesis of animals and plants. For animals, what happens? That it starts at the side and then it moves towards the center, pinching of the two cells separately. And that happens by furrow formation. Furrow means deepening. And in the case of plants, it would start at the center and then move towards the periphery. Now, the formation of new cell wall begins with the formation of a simple precursor called the cell plate that represents the middle lamella between the walls of two adjacent cells. So, in the animals, it takes place by furrow formation. In plants, it takes place by cell plate formation. Now, at the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium. Example, liquid endosperm in the coconut. The watery part of the coconut, that is a result of karyokinesis not being followed by cytokinesis, and that is what we call as syncytium, which is a multinucleated condition. Do you people remember where is this condition found other than this liquid endosperm? Any idea? Hmm? Girls respond, please. Say yes or no. Even if you don't know, say no, ma'am, we don't know. And also switch on your video because it seems like I'm talking to myself. Please switch on your videos so that I can see you while I'm teaching. Come on, quickly. I have given you the option to switch off, but you people don't respond in the class. So switch on your videos. But I'll know that, okay, you are understanding or not. Come on, quickly.
Yes, ma'am, we understood.
So switch on your videos. Both of you, come on. It's only I can see you. That's it. No one else can see you, nor does your video get recorded. Please switch on your videos.
Ma'am, I cannot switch on my video.
Why? What happened?
I'm not at my place.
Okay, so at least respond.
Yes, ma'am, I will.
So cytokinesis is clear to both of you?
Yes, ma'am.
Moving forward, we'll talk about the significance of mitosis. Why is it important? What is the function and where is it employed? Now, mitosis or equational division is usually restricted to the diploid cells, meaning the cells which have two sets of chromosomes. But in some lower plants and in some special insects, haploid cells also divide by mitosis. So this is an exception to this rule wherein it says that mitosis will only take place in the diploid cells. But for the lower plants and some insects, the haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Now see, haploid ones. Have you studied about the this thing, sex determination and the reproduction in the case of honeybees? Do you people
Yes,
remember
ma'am.
that? Yeah. So for honeybees, you know that the males of the honeybees, they are haploid, isn't it? So those fertile haploid males, they divide by the process of mitosis. Now, mitosis usually results in the production of diploid daughter cells with identical genetic complement. If the parent was 2N, the offspring will be 2N. The growth of multicellular organism is due to mitosis. Any growth that takes place inside the cell or the increasing of the cell, increase in the number of the cell, that also happens with the help of mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. If it therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio, because one cytoplasm will contain one nucleus and that is how it is supposed to happen. Now, if anything changes that, so then mitosis comes into play so that this can be regulated properly. Okay. Now, a very significant contribution of mitosis is cell repair. So up until now, we have seen mitosis is responsible for cell growth, cell repair. Now, the cells of the upper layer of the epidermis, cells of the lining of the gut, and blood cells are constantly replaced. And how are they replaced? They are replaced by mitosis. Mitotic divisions in the meristematic tissues, meaning the apical and the lateral cambium, results in the continuous growth of plants throughout their life. Since the growth in plants is open, it's like indeterminate. We cannot fix the growth of the plant. It keeps on growing. That is because of mitosis it takes place. Now, my question to you is, at the end of mitosis, how many cells are formed or how many daughter cells are formed?
2.
2 daughter cells. What is their ploidy?
Diploid.
Diploid. Moving forward, we are going to discuss about meiosis. Now, meiosis is the reductional division. Also, remember one thing, for those insects that are haploid, mitosis will become their mode of reproduction. Just like how meiosis is employed for reproduction in us, right, for our germ cells, but for haploid cells, it is employed, like mitosis will be the considered for the haploid insect, that their whole body is haploid for those cases. Now, talking about meiosis. Now, production of offspring by sexual reproduction includes the fusion of 2 gametes. Fertilization is necessary, each with a complete haploid set of chromosomes. In order to form a 2N organism, N plus N, or the fusion of a male haploid with a female haploid, has to take place in order to form a diploid cell. This specialized kind of cell that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is known as meiosis. Now remember one thing, that at the end of meiosis, haploid, like 4 haploid cells will be formed, and at the end of mitosis, 2 diploid cells will be formed. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. Very important line. That haploid phase is restored because of meiosis, and the diploid phase is restored because of the fertilization. When 2 haploid gametes fuse, a diploid structure is formed, and that is how it restores the diploid phase in the organism. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. Now, talking about the key features of meiosis. Meiosis involves 2 sequential cycles. Just like how mitosis went in one cycle. All the stages were there and it happened in one cycle. But talking about the meiosis, so meiosis would involve the sequential cycle of nuclear division and cell division. And it is divided into 2 types, meiosis 1 and then meiosis 2 happens. And both of them happens in one cycle only. Now, meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatids at S phase. During the S phase, DNA replication takes place. Now, meiosis involves the pairing of homologous chromosomes and recombination between non-sister chromatids of the homologous chromosome. For example, if I draw, this is one chromosome and this is another chromosome. Now see, these 2 structures, the chromatid, they are the sister chromatids, meaning the chromatids of a same chromosome will be called as sister chromatids. But if you take this one and this one, so they will be non-sister chromatids. So recombination between non-sister chromatid takes place. At the end of meiosis, 4 haploid cells are formed. Now, just like how the division, like mitosis was divided into prophase, metaphase, anaphase, telophase, same way, the meiosis 1 and meiosis 2, both of them are separately divided into those exact phases. Prophase 1, metaphase 1, anaphase 1, telophase 1, same goes for the meiosis 2. Now discussing about the meiosis 1, it is the first half of the meiosis. Now, prophase 1. Now, the prophase 1 of this meiosis 1, it is very long. It is not as like exactly similar to the mitotic prophase. Prophase of the first meiotic division is typically longer and more complex when compared to the prophase of mitosis. It has been further subdivided into the following 5 phases based on chromosomal behavior. Now, what are those 5 phases? Leptotene, zygotene, pachytene, diplotene, and dikinase. This is the sequence and this is how you will remember it. So again, giving you a mnemonic to remember it, you can remember it like this. Leena, Zara, Leena, Zara, pani de do. Leptotene, zygotene, pachytene, then diplotene and dikinase. Okay, this is the mnemonic. If you need it, you can use it. Now talking about the leptotene stage, what happens here? So during leptotene stage, the chromosomes become gradually visible under the light microscope. Meaning compaction, decondensation of chromosome is taking place. Now the compaction of chromosomes continues throughout the leptotene. Initially, how is the chromosome present in the nucleus? What is the state of the chromosome? Hmm? Tell me, girls, what is the state of, like initially if you look at a cell and if you look at the nucleus of the cell, what will be the state of the chromosome?
Thread-like structures.
Thread-like structures, okay. But in which form? कैसे situation में you'll find it?
Ma'am, didn't understand your question.
See, if you look at a normal cell, okay, and you look at the nucleus of that cell, and inside you'll find the chromosome. In which situation or in which form would the chromosomes be visible to you? You remember I told you something like this? Tangled ball. You cannot find the end, where it starts, where it ends when you look at the normal chromosome. So during this leptotene stage, it starts untangling. Compaction starts taking place, and because of which now the chromosomes can be seen like this. You can actually distinguish out the chromosomes. Now, after leptotene ends, meaning the compaction happens, then it will be followed by the second stage of prophase 1, that is known as zygotene. Now, during this stage, chromosomes start pairing together, and this process of association is known as synapsis. Now, pairing of chromosome happens. Okay, and when pairing of chromosome happens, it makes a homologous chromosome. Each of these are the homologous chromosomes. Such paired chromosomes are called as homologous chromosomes. Homo means same. Now, electron micrographs of this stage indicate that chromosome synapsis is accompanied by the formation of a complex structure called as synaptonemal complex. See, this is a proteinaceous complex which is there to stabilize the pairing. Okay. Now, the complex formed by a pair of synapse chromosomes is known as a bivalent or a tetrad. However, these are more clearly visible at the next stage. So, this is one homologous chromosome, this is another homologous chromosome, and in between them, a synaptonemal complex is there. And then since 1, 2, 3, 4, 4 chromatids are there, so we'll call it as a tetrad or a bivalent. Now, the first 2 stages of prophase 1 are relatively short-lived compared to the next stage, that is pachytene or pachytene. During this stage, the 4 chromatids of each bivalent chromosome become distinct and clearly appear as tetrads. Now, this becomes like clearly visible so as to observe what is happening in the nucleus. Now, this stage is characterized by the appearance of a recombination nodule. Now, see, as I told you that during the meiosis 1, there will be recombination taking place. Now, what is recombination? Do you people know? recombination? Any idea what is recombination? See, recombination means the exchange of genetic material. When, for example, if this is one non-sister chromatid and this is one non-sister chromatid, and when they will be attached in such a way that one part of this chromatid gets exchanged to this and vice versa, then that is known as recombination. It is the exchange of genetic material. Clear? Recombination is clear?
Yes, ma'am.
Now, this recombination or exchange of genetic material can also be called as crossing over, because literally a cross is made. Now, the sites at which crossing over occurs between the non-sister chromatids of the homologous chromosome. At that point of time, see, for example, if this is the homologous chromosome, so this is one chromosome, and same way this is another chromosome. Okay, so see, this is the point wherein the exchange of genetic material is taking place. So at this point, this circular structure is the nodule, which is known as the recombination nodule. And this is the point where the exchange or the crossing over is taking. Now, crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme mediated process, and the enzyme involved is called as recombinase, meaning because of the presence or action of this enzyme, the recombination will take. Now, crossing over leads to recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of Pkt, leaving the chromosomes linked at the site of crossing over, meaning at the recombination nodule, they will be linked together after the Pkatine ends. Now coming on to the next stage, that is diplotin. So the beginning of diplotin is recognized by the dissolution of the synaptonemal complex. And always remember there was a protein complex in between them, which was synaptonemal complex. Now during the diplotin stage, this protein complex will dissolve, will be removed, but they will still be attached at this particular point. And the tendency of the recombined homologous chromosome of the bivalents to separate from each other except at the site of crossing over. The two homologous chromosomes will separate like this, but it will not move from the part wherein the crossing over has taken place. The X-shaped structures are called as chiasmata. Now, this structure will be known as chiasmata. Now, this is an exceptional example that in the oocytes, the cells that are responsible for the formation of the egg of some of the vertebrates, diplotin can last for months or years, meaning during the cell division, during the mitotic division, their cell can stay at this particular stage, diplotin stage, for months, several months or years. After that comes the last stage of prophase 1, that is dikinesis. Now this is marked by the terminalization of chiasmata. Now this part will get terminalized, removed. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosome for separation. By the end of dikinesis, nucleolus disappears, nuclear envelope disappears, all the cell organelles will start to disappear. Dikinesis represents the transition to metaphase. Now, once the dikinesis is done, meaning the prophase 1 has been completed. And normally, for the normal prophase, which happens at mitosis, what we find? We find that the cell does not have the nuclear envelope. It does not have the membrane-bound nucleus such as mitochondria, Golgi, ER. None of that is there. So same happens here also in the prophase 1 of meiosis. At the end of dikinesis, nothing can be seen. Nuclear envelope also is invisible. Now, coming on to the metaphase. Now, we know that meta means second, right? So, it is the second phase. Now, the bivalent chromosomes align on the equatorial plate. the microtubules from the opposite poles of the spindle attached to the kinetochores of the homologous chromosomes. Now, in that case, the single chromosomes were aligned at the metaphase in the case of mitosis, but here the bivalents, meaning the tetrads, will be aligned at the equatorial part, that is metaphase. Now moving on to anaphase. Ana means up. Now the homologous chromosomes separate while sister chromatids remain associated at their centromeres. See, in the mitosis, splitting of centromere took place in the anaphase, but here the centromere is not splitting yet. The chromosomes are going to the opposite pole. In that case, chromatids went, meaning one part of the chromosome went to the other pole and the other part went to the other pole. But here, the whole entire chromosome is going to the different poles. Clear? This much is clear?
Yes, ma'am.
Any doubts up until now?
No, ma'am.
Okay. If you do have any doubts, please let me know. Now moving forward to the last stage of meiosis 1, that is telophase 1. Now the nuclear membrane and the nucleolus reappear, just like how it did in mitosis. Then cytokinesis follows, and this is known as dyad of cells. Although in many cases, the chromosomes do not undergo dispersion. They do not reach the extremely extended state of interphase nucleus. So after the meiosis 1 is done, then there is the need of again a preparatory phase. So the stage between two meiotic division is known as interkinesis. It is a short period wherein the preparation happens. But one thing to remember, that during the interphase, there is no replication of DNA. Meaning, once the meiosis has started, before that the whole entire G1, S, G2 will take place. So in that point of time, what happens? That is the interphase, and there the DNA replication will take place. But during the interkinesis, meaning between M phase, the meiosis 1 and meiosis 2, in between that, the stages there, that is interkinesis, and during that point of stage, there will be no DNA replication. Interkinesis is followed by prophase 2, a much simpler prophase than prophase 1. Now see, the cells which are formed at the end of meiosis 1, this is one cell and this is two cell. Now both of these cells will undergo the meiosis 2, these two only. Now talking about the meiosis 2, again the prophase, metaphase, telophase, anaphase, telophase, all of this will be here. But here it will be much simpler. Now, meiosis 2 is initiated immediately after cell division or cytokinesis, usually before the chromosomes have fully elongated. Meaning, meiosis 2 starts before the chromosome starts getting tangled once again. So, in contrast to meiosis 1, meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 1. The chromosomes again become compact. Later on, metaphase happens. Now at this stage, the chromosomes align themselves at the equator and the microtubules from the opposite poles of the spindle get attached to the kinetophore. So again, a metaphasic plate will be formed. Then comes anaphase 2. It begins with simultaneous splitting of centromere. Now the centromere is splitting now. Right? Because centromere is the point that is holding the two chromatids together. Now, when the centromere would split, the two chromatids will also split. And then because of that point, the opposite poles get the sister chromatids. And then how does they get it? By the shortening of the microtubules attached to the kinetochore. Because if this was the chromosome attached by the centromere and microtubules came here, now this microtubule would start shortening itself, because of which the centromere would split and the chromatids will move to opposite poles. See this thing. How? Initially, this will go back here, this will come back here. So that is what is happening. Now, at the end of the meiosis 1, that is the telophase, meiosis ends with telophase 2, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis is following that particular thing, resulting in the formation of tetrad of cells. Tetrad means 4, and 4 haploid cells are formed at the end of meiosis 1. So if like a very common question that does come from this particular part is that the splitting of centromere takes place at which stage and in which type of division, meiosis 1, meiosis 2. So you should know that the splitting of centromere takes place in the meiosis 2, not the meiosis 1. There is no DNA replication in the interkinase. Now talking about the significance of meiosis. Now meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across generations in sexually reproducing organisms, even though the process per se paradoxically results in the reduction of chromosome number by half. It also increases the genetic variability in the population of organisms from one generation to next. Variations are very important for the process of evolution. Now, you people have studied evolution, right?
Yes,
Hmm?
ma'am.
Yes.
Now, do you know about variations?
Yes.
Can you describe variation quickly? What is variation?
Uh, Ma'am, variation is the change in the organism in
Hmm.
order to adapt better into the environment.
Okay. Okay, good. You're partly right, but how does that change happen? By the introduction of new characters. See, variation means introduction of new characters. That is why sexual reproduction is so much more advantageous as compared to the asexual one, because here the fusion of gametes take place. And when gametes fuse, so some characters from the paternal side, some characters from the maternal side, both of it is coming, and the organism is getting a new variety of characters. So when meiosis is done, always remember the variations are introduced because of meiosis. Clear? Now, can you tell me how and at which point? It's there in the chapter only. I have taught you. So tell me at which point variations are introduced. Hmm? Variations, meaning the introduction of new characters, happen at which point in meiosis? It's very, very easy. Try to answer.
Mitosis.
No, no, no. It happens in meiosis only, not mitosis. Hmm? See, do you remember that point wherein the exchange of genetic material was taking place? Hmm? Crossing over. अभी तो पढ़ा है नहीं नहीं
Yes,
समझ में आया
Professor.
हाँ prophase 1. So at that point of time, this crossing over, which is the exchange of genetic material, that brings about the variation. This is the reason for variation. Clear?
Yes, ma'am.
So with this chapter is over. Now tell me if you have any doubts. I'll give you 2 minutes, no, 3 minutes. Just go through the chapter, just roughly go through the chapter and tell me if is there anything that you do not understand. I'll tell you once again. Or if there is nothing, then I'll start a new chapter. Hmm? Next chapter is photosynthesis. The significance of meiosis is clear, students?
Yes, ma'am.
Yes.
Okay. Are you going through the chapter or am I disturbing in between? Just go through it once. I'll wait. I'll keep quiet for a while and you tell me if you have any doubts. Yes, any doubts?
No, ma'am.
No, ma'am.
Sure. This whole chapter is clear. I
Yes,
mean,
ma'am.
this chapter is easy only. The questions that come from this chapter are also very easy. ये तो उतना difficult chapter है नहीं. So I really hope that there are no doubts. Or just in case later on, if you do have any doubts, you can come with it. Okay. Sure. Shall I move forward then?
Yes.
Yes, ma'am.
All right. All right. So moving forward, we have the next chapter, that is photosynthesis in higher plants. Now everyone knows here what is photosynthesis. तो जल्दी से बता दो क्या है? What is photosynthesis?
Plants preparing food by using chlorophyll.
Huh? Say it again.
The process by which plants prepare their own food using chlorophyll.
Just chlorophyll. Only chlorophyll. Is there any other thing they also use?
Sunlight, air, water.
Speak clearly. I can't hear you. Say it again. What were you saying? Am I audible?
They use sunlight too.
Okay, sunlight. That's it. Or any other thing also?
Carbon dioxide, water.
पूछूंगी तब बताओगे? Yes, the process by which the green plants make their own food by using the carbon dioxide, water in the presence of sunlight and chlorophyll pigment, right? And that is why they are known as autotrophs. So in this particular chapter, we're going to see everything that happens behind the scenes in photosynthesis of higher plants. Now, we know that because of the photosynthesis, the food that we get, it is the primary source, right? Meaning because of the photosynthesis only, we'll get all the different kind of foods that we do get from the plants, right? So all of the animals, including human beings, depend on plants for their food. Just 1 second. Yes. Now, green plants have to make or rather synthesize the food they need, and all the organisms depend on them for their needs. Now, green plants synthesize their food through the process of photosynthesis, and because of this very reason, they are known as autotrophs or photoautotrophs. Now, This process of photosynthesis, it is a physico-chemical process by which they use light energy to drive the synthesis of organic compounds. And where does this light energy come from? The light energy which is used by the plants, where does this come from?
Sun.
Sunlight.
Sun. Very good. From sun. Now see, this light energy that the plant takes in from the sun, it is converted into a chemical energy. And then they do the whole process. Okay, so this is how the basic thing goes behind. The light energy gets converted into chemical energy. Now, all living forms on earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on earth. It is very important due to two reasons. First is, it is the primary source of all the food on earth. Then apart from that, when the photosynthesis is done, during the reaction, there is a reactant, then there is a product, and then there is a byproduct. So what is the byproduct of photosynthesis? It is oxygen. Now this is a byproduct of photosynthesis, but all of the animals, they require it for respiration, for living alive, right? Living alive, for being alive or staying alive. So this oxygen which is released into the atmosphere by the green plants is used by us for the process of breathing. Now, okay. Now see, in the initial part of the chapter, they have explained as to how different components which are used in the photosynthesis, whether it is light, water, chlorophyll, how they are important for plants for photosynthesis. So we're going to see how each of the components are important. Just like the chlorophyll, which is the green pigment, the light, the CO2, all of it is required for the photosynthesis to occur. Now, you may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with a black paper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaves in the presence of light. Why the green parts? Because the green parts of the leaf will only have the chlorophyll present, and chlorophyll is responsible for the light. Like when the light comes from the sun, how does the plant get it? The plant gets it because of the chlorophyll. That chlorophyll is responsible for the trapping of light. Okay, that is why the green parts of the plant can do the process of photosynthesis. Now, there is another experiment in which the potassium hydroxide is used. Now, potassium hydroxide is like, cotton is dipped into it and it is put inside any jar or any place. And what is the function of potassium hydroxide is that it absorbs CO2. Any CO2 that is there in the environment, in the vessel or the environment, will be absorbed by the KOH or potassium hydroxide. Now, if CO2 is not there, then 100% the plant will not make its food. Okay. Now, so if the plant part that is, growing in the KOH rich area or the KOH cotton area, they will not make the starch or the glucose. But the one that does not have KOH present will make the glucose or the starch, and it will test positive for the starch present. Now let us look at the early experiments which were done so as to say that which part is needed and all everything. Now it is interesting to learn about those simple experiments that led to a gradual development in our understanding of photosynthesis. The first proponent, Joseph Priestley, from the year, this is his lifespan. Now, in the year 1770, he performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen. Now, he was the person to discover oxygen. Now, Priestley observed that a candle burning in a closed space, meaning inside a bell jar, it gets distinguished or extinguished, meaning it gets blown out. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air, meaning in a closed vessel which is not in contact with the outside environment. So the animal since it takes in oxygen and gives out carbon dioxide, meaning now oxygen is not there in the surroundings, so it is just giving out carbon dioxide and ultimately taking in carbon dioxide only, so it will die. Same goes for flame. The fire will burn when oxygen is present in the environment. If oxygen is not present, the fire will not burn, so the candle gets blown out or extinguished. Now, what he did later on is that he put a mint plant, took a mint plant and put it in the bell jar, and then he found that the mouse was alive and the candle kept on burning. So he said that whatever foul thing that the mouse or the candle was adding into the atmosphere, the plant was purifying it, whatever it was. Initially, he did a hypothesis about that. Now, he said that plants restore to the air whatever breathing animals and burning candles remove. Meaning both of them were adding CO2 in the air, because combustion is resulting into the formation of CO2. Now later on, it was found out that the plant is responsible to give oxygen, because of which the mouse kept, it was alive and the candle kept burning. Now, using a similar setup as one used by Joseph Priestley, but placing it once in dark and once in sunlight, Jan Ingenhouse showed that sunlight is essential to the plant process that somehow purifies the air fouled by the burning candles of breathing animals. Ingenhouse, in an elegant experiment with an aquatic plant, showed that in bright sunlight, small bubbles were formed around the green parts, while in the dark they did not. Later, he identified these bubbles to be of oxygen. So, with this experiment, two things were actually proved. First, that plant gives out oxygen as a byproduct. Secondly, that plant requires sunlight to do this whole entire process that will give out the oxygen. And another thing, that only the green part of the plant that could release oxygen, because the oxygen bubbles were found in the green parts of the plant, only near the green parts. It was not until about 1854 that Julius von Sachs provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. What is the chemical formula for glucose?
C6H12O6.
Yes, and it is the food of plant, right? Now tell me one thing. This glucose, it is stored in the plant. What is the reserve food material of the plant? The reserve food material of plants.
Cellulose?
No, cellulose is the cell wall composition. Like cell wall is made up of cellulose. What is the reserve food material? It starts with an S.
Sucrose?
This starch. Okay. Starch is the reserve food material. Meaning, see, when glucose has been synthesized in plants, not all of it can be used at once. So, it is converted into a structure which is starch to store it. Okay. Do you know what is the reserve food material of animals?
Glycogen.
Very good, glycogen. Now, his later studies show that the green substance in plants, which is the chlorophyll right now, he did not know. He said that it was located in some special body within the plant cell. So his hypothesis was right. He said that the green pigment or the green substance, it is located in some kind of special body. He found that the green parts in plant is where glucose is made, and that though the glucose is usually stored as starch. Now consider the interesting experiments done by T.W. Engelmann. Now using a prism, he splitted light into its spectral components. See, whenever a glass prism is used and light is made to be incident on it, so it splits the light into its 7 spectral components, isn't it? The Vibgyor, right? You might have studied this in physics also. Whenever light is made incident on a prism, it splits the light into its spectral components. That is violet, indigo, blue, green, yellow, orange, and red. And then he illuminated a green alga. He put a green algae over here, and he wanted to maybe find out that where is, in which region of the spectrum of light is the photosynthesis taking place. So the algae he used was cladophora. Now, okay, one more thing to remember here is that talking about T.W. Engelmann, he used cladophora. But talking about the first person, Jan Inge's house, he took mint plant. So remember which human used which plant for his experiment. Now, so he took cladophora, which is a green algae, and he placed it in a suspension of aerobic bacteria. The bacteria were used to detect the sites of oxygen evolution, meaning the place where the bacteria would accumulate would resemble or denote that at that point oxygen is being released. So he observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. In this part, in the red light and the blue light. Only in these two areas of the light, the bacteria accumulated. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectra of chlorophyll A and chlorophyll B. See, chlorophyll is of many types: chlorophyll A, chlorophyll B, and chlorophyll C. These are the types of chlorophyll. And always remember that these pigments work in the blue and the red light area of the light, not the green. Mostly we think that, okay, since green plants are responsible and everything, so we'll like the process will take place in the green light. No, it takes place in the blue red light. Now, by the middle of the 19th century, the key features of plant photosynthesis were known, namely that plants could use light energy to make carbohydrates from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen was like this: CO2 plus H2O in the presence of light, also chlorophyll, gives us glucose and oxygen as a byproduct. Now this is like the CH2O, it represents the base formula for glucose, that is C6H12. Now a milestone contribution to understanding of the photosynthesis was made by a microbiologist, Cornelius van Niel, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light dependent reaction because it takes place in the presence of light, in which hydrogen from a suitable oxidizable compound reduces the CO2 to carbohydrates. Now this can be explained like this. In green plants, the hydrogen is coming from the water. So H2O is the hydrogen donor and it gets oxidized to oxygen. See, when you split a water molecule, what will you get? You will get H2, H plus O2, right? This is what you'll get. Hydrogen you'll get and you'll get oxygen, because when you combine H and O2, you'll get H2O, like two molecules of hydrogen and one molecule of oxygen will give you the water molecule. So when you will split the water molecule, you will get the hydrogen and the oxygen. So this oxygen is coming from the water also and the hydrogen as well. Some organisms do not release oxygen during photosynthesis. When H2S, hydrogen sulfide, was used instead as the hydrogen donor for purple and green sulfur bacteria, the oxidation product is sulfur and or sulfate depending on the organism and not O2, right? So he said that the oxygen which is evolved by the green plant as a byproduct, it comes from the water and not from the CO2. So always remember, the oxygen which is released as a byproduct in photosynthesis, it comes by the splitting of water. Clear? So this is, and another thing, that when water splits, hydrogen ion is released and that hydrogen ion is used to actually reduce the CO2 into the glucose molecule. The O2 released is from water. This was proved using radioisotope techniques. Note that this is not a single reaction, but it takes place in a multi-step process, which is known as photosynthesis. So the early experiments are clear to you people?
Yes, ma'am.
Any doubts?
No, ma'am.
Sure.
Yes.
Now moving forward, we'll talk about the next topic that says, where does photosynthesis take place? Now we know it takes place in the green parts of the plant because the green parts of the plant contain the chlorophyll. But the chlorophylls are present inside the chloroplast. So we'll see all of these things. So of course our answer would be in the green leaf or in the chloroplast. Now you are definitely right. Photosynthesis does take place in the green leaves of plant, but it does so also in other green parts of the plant. Any green part in plant can perform photosynthesis. Why? Because it has the chlorophyll molecule present. Now you would recollect from previous unit that the mesophyll cell, okay, when we were talking about the anatomy of flowering plants, what are the two types of cells found in the leaves? Mesophyll and bundle sheath. You people remember that?
Yes.
So mesophyll cells in the leaves, they have a large number of chloroplasts present in them. Usually the chloroplasts align themselves along the walls of the mesophyll cells such that they get optimum quantity of the incident light. So inside the mesophyll, the chlorophyll is present. Now within the chloroplast, there is a membranous system consisting of a grana, the stroma lamellae and the matrix stroma. There is a clear division of labor within the chloroplast. Also, do you remember from the chapter cell, chloroplast or plastid is known as what type of organelle? Chloroplast or the like the plastid in general, they are known as which type of organelle? Do you remember semi-autonomous?
Yes.
No. What is semi-autonomous? Girls, answer. What happened? Answer. सब पीछे का भूलते जा रहे हो? Huh? I try to connect chapters together. Why aren't you people answering? नहीं याद है?
No, ma'am.
Because this is why I asked you to read NCERT. Do you people read NCERT? पढ़ते हो क्लास के बाद NCERT?
मैम क्लास के इमीडिएटली बात तो नहीं बट पढ़ते हैं.
पढ़ते हो तो फिर क्यों नहीं याद है? Cell? हाँ. Now see, you have very less time. बहुत कम टाइम बाकी है. NEET is right around the corner. ऐसे थोड़ी चलेगा कि you're not understanding what semi-autonomous is. ये तो NEET का बहुत famous question है. क्लोरोप्लास्ट के अलावा और किस ऑर्गेनेल को सेमी ऑटोनोमस बोला गया है?
Mitochondria.
हाँ, क्यों है सेमी ऑटोनोमस? बताओ. You're right. Why? Why it is semi autonomous? See, because they have their own DNA, they have their own ribosome. That is why they are known as semi autonomous. Okay. ऐसे नहीं चलेगा. You have to keep on revising or else you'll forget everything NEET के time पे. If you constantly keep on revising and answering the questions, then only you're like this discussing, like I discuss the NCERT PDF. Why do I do so? मैं तो अपने notes use कर सकती हूँ ना. I use this so that you get a clear cut line of what NCERT is saying to you. है ना? So NCERT reading is very important for NEET exam. Now, the membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar, which in turn forms starch. Now, the first part of the photosynthesis that is responsible for absorbing the light, okay, and that happens directly under the light. That type of reaction is known as light reaction, and the other name for light reaction is photochemical reaction. The ones that are not directly dependent on the light, but are dependent on the products of the light. See, after light reaction is done, ATP and NADPH are formed. Now the dark reaction, which is the second type, it is dependent on ATP and NADPH. So we'll call it as dark reaction or the carbon reactions, because they are not directly driven by the light, but they are dependent on the products of light reaction. But it does not mean that the dark reaction occur in the dark. Okay. They are dependent on the light, but not directly dependent. This much is clear. Like, where does photosynthesis take place is clear to you?
Yes.
Okay. Moving forward, we'll talk about the different types of pigments that are involved in the process of photosynthesis. Now, looking at plants, have you ever wondered why and how there are so many shades of green in their leaves? Even in the same plant, you'll find multiple shades of the green color. Now, why is it there? Because there are many components of pigment that are green, yellowish green, light green, bright green, all sorts of colors or shades are there. And how do we get to see them, and how do we get to separate those pigments? By the use of paper chromatography. A chromatographic separation of the leaf pigments show that the color that we see in leaves is not due to a single pigment, but due to four pigments. Like, the four major pigments which are found in the leaves are chlorophyll A, which is bright or blue-green color in the chromatogram. It is bright or blue-green. Then there is chlorophyll B, which is yellowish green, xanthophylls, which are yellow in color, and carotenoids that are yellow to yellow-orange. Now, see, what is the function of the presence of pigment? Pigments are responsible for absorption of light at different wavelengths. Now, let us study the graph showing the ability of chlorophyll A pigment to absorb light of different wavelengths. Now, we know that light has several spectral components, and these pigments are responsible for absorption of light from those spectral components. So when we find out the spectrum of the chlorophyll A, it comes out to be the highest of all the other pigments. And it also tells us that the chlorophyll A shows the maximum absorption. It is the most efficient one. Okay, now maximum absorption is by chlorophyll A, that is in the blue and the red region. And it, like the chlorophyll A, because of this maximum absorption, it shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. Always remember, the chief pigment will be chlorophyll A. Now, what about the other pigments? So those are known as the accessory pigments. See, these graphs together show that most of the photosynthesis takes place in the blue and red regions of the spectrum. Some photosynthesis does take place at other wavelengths of the visible spectrum. Let us see how that happens. The chlorophyll is the major pigment which is responsible for trapping light. Other thylakoid pigments such as chlorophyll B, xanthophylls, and carotenoids, these three pigments, the other left three pigments, they are known as the accessory pigments. They also absorb light and transfer the energy to chlorophyll A. Okay, their function is also absorption of light, but once they absorb the light, they transfer the energy to chlorophyll A molecule. So apart from that, another function that these accessory pigments perform is to protect the chlorophyll A from photooxidation, meaning from the chlorophyll A to get destroyed. It is protected by the accessory pigments. So to absorb light from the various wavelengths and to protect the chlorophyll A, that is the function of accessory pigment. It is not the chief pigment. Chief pigment is just chlorophyll A, rest all are accessory pigments. So the pigments are clear?
Yes, ma'am.
Any doubts?
No.
Okay. All right, moving forward, let us talk about the light reaction. Now, light reaction is the one that happens obviously under the sunlight, and as a result of which, what happens? ATP and NADPH are synthesized. Also, the whole process of absorption of light energy, converting it into chemical energy, splitting of light, everything happens. So light reaction, they are also known as photochemical phase. It includes the absorption of light, water splitting, oxygen release, and the formation of high energy chemical intermediates that are ATP and NADPH. These are the following events that will take place under the light reaction. Splitting of light, light absorption, splitting of light, oxygen release, formation of the high energy chemical intermediates, that is ATP and NADPH. Several protein complexes that are involved in the process. Okay, these proteins are organized into the light harvesting complexes. Right, so see, there are complexes in which the proteins are arranged into, and there are photosystems, photosystem 1 and photosystem 2. Photosystem 1 was discovered first, so it was named as photosystem 1. The next one was discovered later. Now, the light harvesting complex are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system also called as antenna. See, what happens is that chlorophyll A, since being a chief pigment, it has to be present separately. Okay, it has to be present separately so that it can absorb the maximum amount of light. Now, these pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystems. The PS1, that is the photosystem 1, the reaction center in the photosystem 1 has the 700 nanometer absorption peak, meaning in the reaction center of the PS1, the chlorophyll A will absorb the light at 700 nanometers. That is why the PS1 is also known as P700. While in PS2, it has absorption maxima at 680 nanometer, and this is why PS2 is known as P680. The PS1 is P700. Up till now, is everything clear?
Yes.
Any doubts?
No.
Brain saturate हो गया कि अभी still things can go inside? Are you people tired?
Yes.
Tired? Shall I stop here? Shall I continue?
Stop.
Stop. यहाँ तक सब कुछ समझ में आ गया?
जी मैम.
Sure? 100%?
Yes.
ओके, चलो फिर यहीं तक रखते हैं, क्योंकि जब दिमाग में फिर नहीं जाएगा तो कोई फायदा नहीं है ना? अप until now, have you understood every single thing?
Yes.
Sure, कोई डाउट तो नहीं है पक्का?
Yes ma'am.
Okay, okay, very good. All right, so then let's just keep it till here only. Now in the next class, we'll continue with the electron transport, how that happens and everything. Okay, we'll start from here. If you have any doubts, you can ask me. Do you have any doubts?
No, ma'am.
No ma'am.